Hello everyone, myself Amudalakshmi, working as Assistant Professor in the Department of Biotechnology, Kamaraj College of Engineering Technology. Today we are going to see about the prokaryotic DNA replication, enzymes involved and its significance. Let's get into the video and let's have a brief introduction about a prokaryotic DNA replication. Prokaryotic DNA replication is a process by which the DNA duplicates itself and that copy of DNA is passed on to the daughter cells. Replication occurs before a cell divides because the exact copy of the DNA is divided among the two cells and that lineage has to be maintained. DNA replication uses a semi-conservative model of replication. As we know earlier, we have three types of DNA replication, but here the semi-conservative model is mostly followed. DNA replication uses semi-conservative model of method that results in double-stranded DNA separating into a one parental strand and another daughter strand. Prokaryotic DNA replication is often studied using E. coli as a model. Since it divides in within 20 minutes, it is easy to study. And this is the overview image of a DNA replication. The two original DNA strands will separate. DNA polymerase enzyme adds complementary nucleotides to each strand, and these two DNA molecules forms two identical strands of DNA later. And let's go through the features of DNA replication. The first point we need to discuss and keep in mind is whenever DNA replication starts, whether it may be a prokaryotic or eukaryotic cell, it always starts with the origin of replication. It is also called as ORI site, or it is also called as ORIC site. This process occurs in the cytoplasm of the cell. Since it is a prokaryotic in nature, there is no defined boundary between the nucleus and the cytoplasm. So, mostly the DNA replication in prokaryotic cells carry out in the cytoplasm of the cell. And next point we have to remember is DNA synthesis always takes place in the 3' to 5' direction by a DNA polymerase enzyme, which results in two strands, a leading strand and a lagging strand. The leading strand, which I told earlier, is synthesized continuously, whereas lagging strand is synthesized as a fragment, which we are familiar with the term called as Okazaki fragments. And these are the list of enzymes which involved in the DNA replication process. The first and foremost enzyme is helicase enzyme, which helps in the unwinding of DNA at the origin of replication site. Next one is single strand binding protein, which helps to maintain the separation of the double stranded strand, which helps to prevent the single stranded binding protein helps to prevent the rebinding of double stranded DNA. Next important enzyme is DNA polymerase, which are of three types in prokaryotes, 1, 2 and 3, of which DNA polymerase 3 helps in the extension of DNA in both leading as well as lagging strand. The DNA polymerase 1 is responsible for the removal of RNA primers and substitute it with the DNA nucleotides, and DNA topoisomerase 1 and 2 helps in prevention of breakage of the DNA to overwinding. DNA topoisomerase 1 enzyme creates a temporary break in a single stranded of DNA, whereas topoisomerase 2 creates double break in the DNA strand to prevent overwinding and breaking of DNA. One more enzyme which we miss is DNA polymerase 2, which helps in the proofreading of the DNA once the replication is completed. And last one is DNA ligase enzyme. As usual, we know ligase is for sealing of breaks. Here it is used for the joining of DNA fragments. It works by the principle of forming a phosphodiester bond between the nucleotides. And this is the overview of the picture I want to explain here. This is the original DNA template of the DNA. The helicase enzyme first unwinds the DNA, and the single stranded binding protein helps to maintain this separation. Then DNA polymerase 3 enzyme will come and bind, and it adds nucleotides continuously in leading strand, whereas in lagging strand more number of RNA primers is added since the enzyme works in the reverse direction. After that the RNA primers are removed by primase enzyme and it is sealed by DNA ligase enzyme finally. And these are the detailed steps which I explained before and I have listed one by one. Step 1 is helicase action, which helps in the unwinding of DNA. Step 2 is stabilization of by SSB protein. SSB protein helps to prevent the unwinding of DNA. Step 3 is primer synthesis by primase enzyme. Step 3 is primer synthesis by primase enzyme. It adds RNA primer to the strands so that they can continue with the replication. Step 4 is elongation by DNA polymerase 3, which adds nucleotides to the growing strands of DNA both in leading as well as in lagging strand. Step 5 is the RNA primer previously added is removed by our DNA polymerase 1 enzyme and it is replaced with the DNA nucleotides. And step 6 is 6 and 7 involves the enzyme topoisomerase 1 and topoisomerase 2, prevents the overwinding of DNA by creating a single nick or a double nick respectively. And in step 8, it is segment joining by DNA ligase enzyme, which is actually the formation of phosphodiester bond between the two nucleotides and it seals the gap. And it is one thing after the whole DNA replication process is DNA repairing. It is one of the most important process because this activity proof is also called as proofreading activity. It helps in the conservation of DNA from one generation to another. DNA polymerase 1, 2 and 3 which are present in the prokaryotes will perform this activity, DNA proofing activity, but in eukaryotes we have different sets of enzyme for doing proofreading activity. What does proofreading do is it will check each and every nucleotide whether it is correctly paired or correctly synthesized like that. When a mismatch is detected by this enzyme, the polymerase passes and it reverses back and it cures the mismatch and proceeds along. So, after excision actually polymerase enzyme can excise the mismatched nucleotide and replaces it with the correct nucleotide and it involves DNA ligase to seal it again. And the significance of this proofreading activity is, as I said before, it is very important to maintain the integrity of DNA from generation to generation. So, proofreading activity is very significant and it is carried out by all the 3 DNA polymerase enzymes in prokaryotes with the 3' to 5' exonuclease activity. So, whenever a nucleotide is mismatchedly paired, it halts for a second, it reverts back, it removes or excises the nucleotide and pairs with the correct one. This term is otherwise called as fidelity of DNA. In simple terms, you see here high fidelity is low error rate during DNA replication. This is very crucially important in maintaining the integrity of an organism. Thank you so much.